So, can I start? Yeah, my name is Antonio. I am a PhD student from the bioinformatics program of Boston University, and I'll talk about my research on ChIP-seq data. It's basically a method to identify binding sites with high resolution and also to identify cooperative interactions. Just giving a quick overview about ChIP-seq. It's a technique to identify protein DNA binding. It starts with a DNA. It has multiple binding sites. Some of them are bound or not. We purify this DNA from a cell, and we shear it in many fragments. And in order to identify, purify the fragments that are bound by a transition factor, we use an antibody selective to the query transition factor that will provide a sequence where the binding sites are bound. In order to identify them, we sequence and then align to a genome that will provide a coverage show in this y-x at each genome position. The regions surround the regions that contain a binding site are exemplified by these red bars in this plot. They show an enriched coverage when compared to the background shown by the blue lines in this plot. So just to start, why ChIP-seq? One example, our lab has been working using ChIP-seq to map all the regulatory network of Mycobacterium tuberculosis. Tuberculosis is about 200 transition factors. Also, ChIP-seq is useful to study enhancer, as we saw in the keynote talk today. So the current state of ChIP-seq analysis, there are multiple tools. Most of them are developed to identify binding regions. However, little has been explored to understand what occurs inside each of the enriched regions. So the opportunities that we're exploring in this research, we are using, we want to identify binding sites at high resolution and also cooperative interactions between binding sites. Starting with the high resolution problem, the output of a peak color provide a summit that might seem to be one binding site, but if you look carefully, it might actually contain multiple binding sites in that region. The importance of resolution can be exemplified by the fact that the position, the number of sites, matter to define if a gene is going to be expressed or not, and also to identify cooperative interaction. We model the observed ChIP-seq data as a signaling process. In this case, each binding site represented by this brown box. It is a source of impulse signal, and that emits an impulse response that is observed in the ChIP-seq coverage. The coverage is sequence is strand specific, and then you can see a ChIP-seq, a peak shift in the coverage of the forward and the reverse strand. In the case when you have multiple sites in the same region, we'll observe an overlap, some of the impulse response from these two binding sites. Going very quick, we have a physical model for the impulse response. It basically, the shearing step, we should look at this black box. The shearing step causes many breaking points in the DNA, and after the immunoprecipitation selection, only the break point that is closest to the transition factor is selected for sequencing, which indicates an extreme value problem. The decoding process, we use a blind deconvolution algorithm. It basically, we provide an initial guess of the shape of the impulse response, shown by this solid line, and an initial guess about the position of the binding sites. We reiterate this algorithm, updating the location and also the shape of the impulse response until it reaches convergence. In the first round, we use the predicted binding sites to run a motive search and feed back a new deconvolution process where the search space is constrained by the output of the motive search. The integration of ChIP-seq coverage with motive search, it takes advantage of the high resolution provided by the sequence, and at the same time, it takes advantage of the ChIP-seq coverage to filter out potential false positives. In this figure, I show there are regions that is validated to contain 2 binding sites, and when you run motive discovery, depending on the parameter, you can find multiple potential binding sites. And after our deconvolution algorithm, we are able to predict correctly the binding sites that are responsible for the binding. To validate our results, we use as a reference a set of 47 binding sites that was empirically validated with DNA footprint. Basically, we show how our method is able to boost resolution, the output of peak colors in this first figure, using the summit of output from peak colors is represented by these blue bars, and the fraction of sites detected is shown on the y-axis. When you use our model, we are able to identify most of the sites that are missed by the peak colors. In addition, on the right plot, the whisker plot shows our method provides binding sites at high resolution. The fact that our method is able to filter out false positives is shown in this table. If you use a motive discovery algorithm, depending on the threshold that you choose, you might include more false positives, getting more true positives, but at the same time, you would increase false positives. But if you use a more conservative threshold, you miss some of the true positives. In our method, we are able to increase the number of true positives. At the same time, we filter out for false positives. In addition, when you apply it to eukaryote data, it shows high reproducibility. These figures, it shows the magnitude of the impulse response predict using GABP. Each X is one of the replicates. It shows a high correlation. And also the predicted binding, the distance between replicates, about 75% is within one single nucleotide resolution. Now, let's talk about the case of cooperative interaction. The importance of cooperative interaction, it affects the dynamic of binding. When you don't have cooperative interaction, the probability of binding goes smoothly with the transition factor concentration. And in case of when you have cooperative interaction, you can have a transition close to a true state. And also, when you consider cooperative interaction, it shows to improve models that predict gene expression. So the information to detect cooperative interaction is represented by this pink box in this figure. We model a case that DNA fragments contain 2 sites bound at the same time. In this case, we would have 2 potential impulse response, one for DNA fragments that contain only 1 bound transition factor, and also when there are 2 bound sites at the same time, we call them single and double binding signals. So when the cooperative interaction, it indicates that the bind to one site, to both sites are not independently from the bind of each site individually. So in a toy model with 2 possible configurations, 2 sites, you can have 4 cases where none of them are bound and all of them are bound. And each of those conformations is able to emit a different source of impulse signal. We are able to take from the output of this data an estimator of cooperative interaction. For reference, we use as this data published by Shauhan et al. It shows positive cooperative interaction. And and use as a control data a sum of two sets containing only one site that would be independently, they would have negative cooperativity. And when you plot in this, we show in this plot the data that has positive cooperative interaction. It can be discriminated from our negative control, and when we plot a ROC curve, it shows a curve with an area of 0.75. And at the end, just give acknowledge for my lab, for people who helped me in this research, and thanks for RECOMB for the opportunity of talking here. That's it.
One quick question. So I totally the utility of your method for weeding out false positives. Have you looked at whether any cheap qPCR positives are also being excluded, things that you would throw out, but empirical validation methods would keep in?
No, I haven't looked at that data. But good idea. Yeah.
Have you seen if there are footprints when you see double binding, two double DNase footprints on the regions you find double binding events or you predict double binding events?
Yeah, we actually didn't perform the DNA footprint, so I took from the paper. So okay. What they show is that when you knock out the cooperative binding domain, it disappears the signal. The footprint gets you don't see the both bound together.
Gets flatter.
Yeah.
Okay.
Yeah, exactly.
Thank you.
Yeah.
Hello, Eugene here from Corey. I've done some fair-seek, and I also see the double peak kind of going in the opposite directions. Would your method work to find the motifs underneath fair-seek peaks, or narrow down the resolution, you think?
Sorry, what kind of?
It's like it's like ChIP-seq without the antibody. So you don't necessarily know what transcription factor is causing the peak.
If there is a motif conserved, then if we run a motif query at the end of the first deconvolution doesn't need the motif, but if there is a motif conserved, then it would be output in the second at the end of the first round of the convolution. We run a motif finding, so we perform this in a high throughput that we don't know the motifs. So if it's something there, it would find.
And how fast does this take to run on an average ChIP-seq on a machine?
At this point, it's not optimized. It's taking about 10 seconds per enriched regions, but I can optimize and get about 100 to 1,000 times faster. But I just don't have the optimized one version in a closed format. Yeah.
Thank you.
Yeah, thanks.
All right, I think all the speakers in this morning session